for all 4 presenters will present in the group module, right? Am I correct?
Yes, correct.
Okay, confirm, ma'am. Okay, any more from Indonesia join us now? Good evening, Mr. Marie. How are you?
Hi, hello everyone. Nice to be here again.
Yes, welcome you again. And okay, your voice and picture is very clear. Thank you. And hello, Dr. Usma. Welcome to our phase number 3 again.
Hello, how are you? Good afternoon, everyone.
Hi, we are great here. Good to see you again. Your voice is very clear. Thank you.
Thank
And
you.
may I check with Malaysia team? Mr. Stella will be the presenter. Oh, not yet. Okay. For the housekeeping rules, so first you enter, please rename yourself, maybe your name and country, and please mute your microphone when you are not speaking, and please use our chat box. If you have question or any concern, please use the chat box. We will try to answer all of your questions, and please raise your hand before you are going to speak. Thank you. I heard from Yara that now we have Dr. Hasa. Excel a bit.
We'll try to contact him first, Tanya Puan.
Hello, Tanya Puan. We are here already.
Okay, we will wait for Mr. Selva.
Yeah.
Okay, I would like to inform you again that you have to moderate your own slide and you have 45 minutes per group, including the short Q&A answer after your presentation. Thank you. Okay, without further ado, I would like to invite my supervisor, Ms. Jintana Siwongsa, to give us some remarks. Thank you.
Thank you, Tanyaporn, and very good afternoon to Dr. Usma, Ma'am Damri, my beloved GLLP colleague. Today, the 3rd phase, but also the 3rd time, the 3rd session of our, I would say that piloting and also sharing the course that you interested to work on and in what kind of adaptation that you have done in your own country. So I think Bu Krishna is in the waiting room, so please accept Bu Krishna. And selamat malam to also Bu Hana. So now we see each other online, right? After we saw each other face to face last week. So very nice to have you on board once again today, Indonesia team. So again, RPHL network is very honored to have the 2, prominent facilitator to navigate the conversation, the sharing session from participants. I didn't need to introduce, but all of you already know both of them today. I have Dr. Usma and I also have Ma'am Damri, both of them from WHO to take lead in moderation the session. I don't know whether should I give the floor to Dr. Usma, who's very far away, or should I give the floor to Damri? So first, so is
Um,
that Damri first or
yeah, I'll
Dr.
speak.
Usma first? So, oh, yes.
So
But I
we're
give the floor to the WHO. Let me put it that way. Thank you very much.
great. Thank you. Thank you, Gintana, and hello, everyone. So Uzma and I have swapped roles for today's session. I will lead and she will provide excellent support. And she's really the expert here. So for today's session. So yes, happy to join. And I don't know, maybe Uzma, if you'd like to say a few words before we get going.
Thank you, Damari, and welcome everyone. This is the third of the session, so I think you know the rules and you know how it's going to happen. Very happy to be here, and as Damari says, I think you're the experts here from whom we're going to learn, and we'll try to put in our 2 bits. So welcome, and we're eager to hear what you have to share. Over to you, Damari.
Great, thanks. So Tanyaporn, are we ready to start? Great. So we'll hand over to our Indonesia team to kick us off with their presentation on biosafety today. Over to you, Dr. Hannah and team.
Thank you, Madam Damari. So let me share our screen. Okay. Can you see the screen, our PowerPoint slide?
Yes, it's clear.
Okay, thank you. Good evening, everyone. We are the Indonesia team, and today we will be presenting about biosafety. This is a very important topic since biosafety ensures the safety of laboratory personnel, the environment, and the community. In this session, we will specifically highlight one key aspect of biosafety, which is biosafety risk assessment. My name is Johanna, and I will begin with the first 4 slides before handing over to my colleague. Before we begin, let me introduce our team members. Myself, Johanna, I'm from Human and Environmental Health sector. Second, Annie, she is from Animal Health sector. The third one, Hannah, she is from Human Health sector. And Evie, she is from Environmental Health sector. Together, we will share our perspective and experience on biosafety. This module actually covers many aspects of biosafety, such as principles, program management, policies and procedures, international guidance, laboratory risk mitigation and design, training, incident management, and waste management. These topics are interconnected, forming the foundation for safe laboratory practices. However, for this 30 minutes presentation, we will focus only on biosafety risk assessment because it is the foundation for implementing all other biosafety measures. The definition of biosafety comes from the Biosafety in Microbiological and Biomedical Laboratories by the U.S. Department of Health and Human Services, SHS, or BMBL, the 5th edition 2009, which is considered the cornerstone of biosafety practice and policy in the U.S. According to the BMBL, National Biosafety Guidelines evolved from the effort of the microbiological and biomedical community to promote 3 key elements, which are the use of safe microbiological practices, appropriate safety equipment, and facility safeguards. The goal of these combined efforts is to reduce the risk of laboratory-acquired infections and to protect not only public health, but also animal health, agriculture, and the environment. In other words, biosafety is not just about protecting laboratory workers, it is also about safeguarding the wider community and ecosystem from potential risks that may originate in the lab. Biosafety risk assessment follows 5 structured steps. The first step is gather information and identify hazards. Second step, evaluate the risk. The third step, develop a risk control strategy. The fourth step, select and implement risk control measures. And the last step, review risk and control measures. These 5 steps provide a systematic approach to ensure laboratory risks are well understood or identified and also effectively managed. To conclude my part, covering the introduction and the overview of risk assessment steps, now I would like to invite my colleague Annie to continue with the next slide, starting from step one of risk assessment. Thank you. Please, Annie.
Thank you, Johanna. In the step one, we gather information. This is a hazard identification. Hazard is a source of danger that has the potential for causing harm. In this step, we must look for a lot of information, consider the laboratory process to be performed and following factors that influence risk. What is the biological agent, SARS-CoV-2 or HPAI or anthrax? What is the laboratory procedure and equipment? How about control measures already in place? How about the facility? How about the personnel and other factors? Next. For step 2. In the step 2, we evaluate the risk. Risk is likelihood and consequences of an undesirable event relates a specific hazard. To evaluate the risk, we use this table based on what situation could lead to potential exposure or release. Maybe like what is spill.
Sorry, Miss Annie. Could you please speak louder?
Okay.
Thank you. Thank you very much.
Spill, aerosol, injury. How likely are this situation to happen? Consequence is severity of an event. What happened if the person was exposed? So the consequences of exposure, we have levels severe, moderate, negligible, and likelihood is a probability of an event occurring. Is it possible that person could be exposed? So risk is combining of consequence and likelihood. For example, if consequence negligible and likelihood is unlikely, the risk is very low. This is how to evaluate the risk. Next. After that, We determine the risk level. What is risk tolerance, acceptable risk, unacceptable risk, how is it determined, and who is it determined by? After that, next slide. Under step 3, we develop a risk control strategy. What is the mitigation consideration? Are resources sufficient to secure and maintain potential risk control measures? Will any condition identified limit the ability to reduce risk? Can the work be done without additional risk control measure? And for next slide, and on the step 4, please welcome my colleague Hannah to continue.
Thank you, Bu Eni. So now we are in the step 4, or select and implement risk control measure. So at this stage, we already know the hazard and the level of the risks. So the next step is to decide what additional control are needed. So this control may include personal protective equipment, biosafety cabinet, and other as appropriate. But when implementing, we should always ask 3 key questions. There are: how likely are the situation to happen? What is the severity of consequences of exposure or release? What existing regulation, policy, and also strategies guide to our decision? On the slide, you can see the risk matrix. The matrix combines the likelihood of an event with the severity of its consequences. So for examples, if the consequences is severe and the likelihood is very high, the risk is unacceptable. It falls in the red zone. If this consequences is moderate, but the likelihood is low, so the risk is manageable, in the green or yellow zone. Our goal is always to reduce risk to level that is considered acceptable. The box below explain the residual risks. This means the level of risks that remain even after control applied. So this might involve adding more control measure and etc. Finally, the critical question is, should the work proceed with the selected level of the risks? If the answer yes, so then activity can continue under the established safety measure. Next slide, please. So here we see the situation of high pathogenic avian influenza in Indonesia, which remain a disease of ongoing concern. From the temporal perspective, the data shown major peaks in June 2024 and December 2024. This is a data from last year. This suggests a seasonal pattern when the outbreak become more frequent or more severe at certain times of the year. From the spatial perspective, the highest concentration is found in the West Java, Banten, and then cluster reported at the Central Java, East Java, North Sumatra, and East Timor. So why is this important? Because risk assessment plays a critical role here. It allows us to detect early warning sign for potential large-scale outbreak. It helps us to targeted prevention and also control measure. It supports more efficient allocation of resources and surveillance priorities. And also it informs stakeholders so that policy and intervention can be made in timely manner. From the biosafety perspective, the situation reminds us several key points. First, laboratory personnel handling of high pathogenic avian influenza samples are at risk, and we must identify and mitigate those risks. Secondly, proper use of PPE and containment facilities is non-negotiable. Third, continuous training is required not only for the safe diagnostic work, but also for safe field investigation. And finally, a strict biosafety practice reduce the chance of pathogen escape or laboratory acquired infection. Next slide. This is a risk assessment for H5N1 or avian influenza viruses. First, H5N1 is classified as a risk group 3 pathogen, which means it is a zoonotic agent with serious potential for human disease and require high level of biosafety precaution. The infectious dose of human is still unknown, which increase the uncertainty and risks when handling the viruses. For source of transmission, there are multiple possible routes: direct contact with infected animal or poultry, handling poultry, carcass, fresh tissue, and others, exposure to contaminated environment, infected viral transport media, or embryo eggs used in the laboratory, and of course, direct handling of the virus is allowed and also fluid. Next, in terms of route transmission, infection can occur if the virus comes contact with the mucosal membrane or through aerosols via inhalation or intranasal exposure. The incubation period is typically 2 until 4 days, which is important to monitor exposed staff or tracing potential transmission. Finally, the virus is sensitive to disinfectant and physical inactive. For examples, 1% sodium hypochlorite, 70% ethanol, formalin, and also iodine-based compound. Next slide, please. So now we'll move to the mitigation, which refers to the measure we put in the place to reduce the control risks when working with pathogenic like H5N1. First, at aerosol generating procedure must be performed in class 2 biosafety cabinet. Second, specimen must be always transported and stored in the tightly sealed container to prevent accidental leaks or exposure. Third, the activities involving diagnostic specimen should be performed in at least BSL2 laboratory. Next is emergency response because incident can do happen. From minor spill inside the BSG, cover the spill with the paper towel, apply disinfectant from the outside, allow enough contact time, then clean and dispose of the towel in biohazard waste. For a major spill outside the BSG, the procedure is more strict. Evacuate the room, close the door, then return with full PPE and clean according to the minor spill procedure. For puncture wound and scratches, the report immediately to the biorisk officer or area manager and clean the wound with antiseptic and soap and running water. For a mucosal splash, rinse immediately with an eye wash or clean water and report to the incident at once. Under mitigation, we also focus on protective measures like using the PPE is very essential. For example, gloves, lab coat, shoe cover, eye protection, and mask and 95 respirator for culture or animal work, and also have protection including access to antiviral vaccination and also regular health surveillance by personnel. Finally, we have administrative control, which are about policy and best practices. Here include for microbiology best practice, necropsy of infected poultry should be done in the certified facilities. For the waste management, must follow strict decontamination protocol, either sterilization, chemical disinfection, or incineration. Training is critical, so all the staff must be trained and on specimen transport, safe BSG use, microbiology technique, emergency procedure, and also competency testing. And lastly, there must be evacuation of control, which is can be conducted annual audit and also assessment. Next slide, please. So let's start. This slide show how we apply the risk assessment and also control measure to specify laboratory procedure. We start with the sample collection. The main hazard here are aerosol exposure during sample handling. So the inherent risk is rated medium to high. To control this, we add facial and also respiratory protection. So with this measure, likelihood of exposure become likely to possible, and then residual risk is reduced to low to medium. Next is viral culture. This is one of the highest risks activities because it forms live viruses. Again, this inherent risk is medium to high. Control measure including here ideally BSO3 facilities with the inward airflow, biosafety cabinet, and also enhanced respiratory protection. After this control, the likelihood of exposure is unlikely and the residual risk is breakdown to the low. For the molecular and serological tests like RT-PCR or HAMHI, the hazard are spill or exposure to infectious material. The risk is medium with additional control such as biosafety cabinet, respiratory and eye protection, good ventilation, risk become unlikely. So the reduce the residual risk is too low. Now for sample accessioning, for example, opening, labeling, and also handling the container, the hazard is include leaks, sharp injuries, or spill. The inherent risk is low to medium. So when we adding the respiratory or eye protection and good ventilation, bring the likelihood to unlikely with the residual risk reduced to very low. Finally, whole genome sequencing. Actually, this technique is no direct hazard. One, the sample is inactivated already by the lysis buffer. The inherent risk is already very low, so we think there's no additional control are needed. So the residual risk is negligible. For the next slide, I will please welcome Effie to continue the slide. Thank you.
Thank you, Bu Hana. Good evening, everyone. I will continue to the step 5, review risk and risk control measure. Risk assessment should be a continuous process and should be performed whenever changes take place, like a personnel, facility, equipment, methods, and regulation. Step 5 reviews the elements covered in step 1. As mentioned previously, this is an ongoing process and should be performed whenever changes occur. If a safety breach or potential exposure occurs, the risk and control measure should be reviewed. For example, reviewing control measure also provides an opportunity to examine whether there are better risk control measure than those you have implemented. If so, you should consider implementing new measure to reduce the risk. An activity to examine biological risk and step to develop control measures. For example, consider PPE for laboratory personnel, safety labels for hazardous material testing standards, and how waste is disposed of. Next slide, please. For the summaries, the key messages discussed in this session, biological risk assessment is crucial to ensure the safe handling of H5N1 samples. The 5 stages: hazard identification, risk characterization, exposure assessment, risk estimation, and risk control provide a structured approach. Effective implementation minimize risk to laboratory personnel, the environment, and the public. Training, monitoring, and ongoing review reinforce a culture of biosafety. Integration with SOPs, PPE, engineering controls, and waste management ensure compliance with national and international biosafety standards. Next slide. Thank you. That's all for Indonesia team for biosafety.
Great. Thank you, Johanna, Annie, Hannah, and Evie. That was a really good job switching between 4 different speakers for a single topic. You kept to time, so thank you very much. Let me open for some immediate reflections from the audience. I'll start with Uzma first. Please use the chat feature if you'd like to put any comments to our speakers, or also raise your hand and we'll call you out in a few minutes. But let me invite Uzma first to give her reflections. Thank you, Uzma.
Thanks, Damari. Well done, Team Indonesia. That came across as a strong team effort. It was an all-girls team, and you coordinated very well to make sure that you covered the chosen component of the module that you had selected, and emphasized aspects of it. What I really appreciated was how you brought in some epidemiological data and linked it to the need for a risk assessment that is that the laboratory needs to do, because that's where the lab is asked to provide that response. So I think the message that at least I received, and I was listening to it as somebody who's participating in the training, would be, just because I'm in the lab doesn't mean that I do not need to continue looking at those biological risks that are happening outside in the environment and prepare accordingly. And if that's the message you wanted to give, it came across pretty clearly from that example, because it would resonate with your participants. So keep doing that. The slide adaptation was good. This module itself, the entire component requires 7 hours of work to be delivered. And when you're delivering it, I think an important consideration, I think it's good to give that input now, is when you're doing a risk assessment module covering anything on risk assessment, a lot of things that you tell in theory, you'll have to probably side by side complement that with an activity section. And I was looking at the guidance as well, which is there for the instructor guide. There are some activities that are in there. And since you're also will be training laboratory leaders, that's an area where you'll have a lot of people who have actually undergone some of those trainings, already understand a lot more about biosafety. So as you become instructors and facilitators, hopefully some of you will be, your training and facilitators understand that when the content of the modules is essentially technical, the message as facilitators in leadership program is beyond technical, because we expect those experts to already know a lot about the technical, but what is the message that they need to take away from it? So I really like that angle of linking it to things outside the lab, but I also want to reinforce here that when you're talking about technical components, the idea is not to reinforce the technical pieces of risk assessment only, but also to reinforce how, as a lab leader, as a manager, this risk assessment would be important and how it will be reinforced within your lab. So I really like how you finished by having a strong culture of biosafety, because that is definitely a leader's job to reinforce that, and how you do it through checks and mechanisms in place. I think I want to end with basically a couple of questions to the team, and it can come back later. Is how were the materials and the instructor guide and the PowerPoint content? How did you find reviewing that content? How representative and how relevant did you find it in preparing for your session today? And how did you try to think about it or not from a One Health perspective? So those are two questions. Whether the content, that resources that are available you found those helpful, you found those relevant, what more can be needed, and you can answer some now and rest later. And how did you come at it from a perspective of health and animal health and environment, as the 4 of you participated in preparing study? Thank you. Over to you.
Thanks, Uzma. So maybe I'll hand over to Johanna and the team, whoever wants to take those 2 questions. We can provide some answers now while we wait for more comments. Thanks.
I think I can go first for the question delivered by Uzma. For the slide developed, we choose the biosafety and then choose the like a pathogen who can cover One Health, because we are come from different sector like animal health and environment, and we choose the one who also can handling by different sector, and we choose avian influenza because in Indonesia still an outbreak in the agriculture and the poultry, And also, we are now struggle with the outbreak in the whiteboard also, and we are from the human side. We are looking for the history of the H5N1 infection. So we decided the H5N1 as our experiences from including integration in the biosafety. So and I think it's much difficult to see, because the biosafety module is very large and very large topic. But we think that the risk assessment is very important as us in the laboratory to how to mitigate the H5N1. And I think additional comment from our college.
Yeah, I just want to add a little part. Yes, as Hannah mentioned, as Hannah explained, we choose this module because we think that these topics is important, especially when we decide what kind of testing we would like to perform in our lab. And it's like a basic assessment that we have to do in our lab. And for the example, yeah, as Hannah mentioned, actually we in Indonesia have about 5 zoonotic diseases, like the priority zoonotic diseases, but we choose this avian influenza. The other ones is like anthrax. Yeah, but we choose this one because many area in Indonesia still have this avian influenza. Yeah, I think that's from me.
Great. Thank you, Hannah and Johanna. One question I had for you before I invite more comments from the audience is, so right now you're all four from the same team, and it's sort of related to the question Uzma asked about how you adapted to a One Health approach when you were deciding on the examples and for the risk assessment. Did you engage with anyone from the environment, animal health sector that has been involved in your GLLP planning process? Thanks.
Do you mean, Damari, when we develop this PowerPoint?
Yes. Sorry, yes. When you were doing this, was there any collaboration across the other stakeholders as you put this together? Give us a sense of how you worked at the country level for this. Thanks.
Since we all four from different sector, Bu Annie from animal, me from human, and Johanna from environment, and also Evie from environment. Before we have an experiences in this assessment of avian influenza, which is also conducted by a different sector of One Health sector in Indonesia. So I think based on that experience, we can develop the model for the GLLP.
Great, thank you. We do encourage that cross collaboration, even as you're adapting the content to the national setting. I'm going to put one of my colleagues on the spot, Paula, who is joining us from our HQ team. I'd like to invite you if you would like to give a few reflections of the presentation today and any questions you may have for the team. Thanks.
Many thanks, Damari, and hello, everyone. Can you hear me well?
We can hear you, but we can't see you. I can see your
Yes.
background, but not your
Yes,
face.
it's quite strange. I'm sorry that you can't see my face, but
Okay.
I'll do a very quick introduction. My name is Paula Gomez. I'm a learning specialist. I've been working with the team, Uzma, Jocelyn, Virginie and others on the design and development of the materials for the GLLP training of trainers. And I wanted to ask to this team to thank you for your presentation and also to ask you a bit more from the learning perspective. What would be the activities that you would recommend in the context as a trainer in the context of the delivery of this module? When I say activities, it may be a group work, an exercise, a guided discussion. I mean, there are many ways to, engage participants into playing with the concepts and with the learning they have done so that they better integrate this learning. So that's my question. What would be the activities you would recommend if you have any ideas to share? Thank you.
Yeah. Thank you, Paula. Miss Damari, is it fine for us to answer?
Yes, please go ahead. We have maybe another 8 minutes left for our session.
Thank you, Miss Paula. Yeah. When we made this PowerPoint, yeah, we think about some exercise for the participant, like we will divide in a 4 or 5 groups, and then each group can choose a topic for doing risk assessment exercise. But sorry, we do not put in the slide because at first we thought that we only allowed to put maximum 10 to 15 slides. That's why we like compact all this information in the 15 slides. Yeah. So we didn't put the exercise, but yeah, we thought that kind of exercise, and maybe we can also make like a quiz, interactive quiz, so it will make the participant more active to participate in this module. Maybe any idea from other colleague?
Yes, an additional Chandra info. Maybe we can have a group discussion, possibly if the participant comes from the different laboratory, like maybe a research laboratory, hospital laboratory, public health laboratory, have different aspect to assess the, for example, H5N1. So I mean the technical laboratory will be different. So the risk assessment will be also different. So maybe we can have a group discussion from possibly different participant laboratory.
Great, thank you. I would encourage all the other participants online to also put in some of your ideas, because I think this question on which activity would you use for this particular module is a great one. So if anybody else has any ideas or some examples, please do share that in the chat with us. Over to you, Uzma.
Yeah, thank you. I think those are great examples that you came up with. So there are a few examples in the instructor guide, and I saw Paula's thumbs up. You've come up with ideas that can go beyond that, and it's great. You wanted to do that, and we understand the constraints of time. But what the idea of the question is to understand whether some of the discussions you had in preparing for those modules included those considerations. This phase 3 is kind of like, before you actually just go and do it for the entire curriculum, you feel confident enough that you actually have all the space and all the freedom to use the content in your perspective to provide the key messages that are need to be given. The other point that I wanted to make, as you were preparing these materials and you've gone through the training in Bangkok earlier, because you are, and correct me if I'm wrong, you're not the participants of a GLLP program per se, right? But you're going to be facilitators of a program. And that may or may not be the case. So the question here is, if somebody is covering biosafety, there are references and connections with other modules in other competencies as well. And those are referenced in that crosswalk of the modules that was, I think, discussed with you in the TOT. It will be very, very important for some of you as facilitators to reference back to those competencies. So at the end, those participants actually understand that. So was at any time during preparation for this module a consideration, or is that something you would think about going forward? Over.
Thank you for your suggestion, Ms. Usma. Yeah, we will put your suggestion to when we become the facilitator.
Great.
And,
yeah.
And may I comment to Mr. Sawang in
I was just going to raise his comment. Yes, please go ahead.
Yeah. Thank you about the word unnegotiable for risk assessment and control. Yeah. And then Mr. Sawang suggests us to present the hierarchy of control in the lecture. Yeah, we agree. And as I mentioned before, since we were told that the maximum only about 10 and 15, so we didn't put the hierarchy of control, but great suggestion. We will put in our slide when we became facilitator. Thank you, Mr. Sawang.
Great, thanks. Let me turn to Gintana and the team for any comments before I summarize the session. Thank you.
Namri?
Yes, Gintana.
I really appreciate, the presentation from Indonesia. I think what they done is very good practice. They really share what they adapt from the main curriculum that they put it in the module. So very good point, very good point. What I like the most is the profile of the first page. Very nice one. Thank you very much. Well done, the Indonesian group. Thank you very much.
Thank you, Ms. Gintana.
Thanks, Gintana. Okay, so I just will quickly summarize what we've heard today. I think it was a very good team effort, as highlighted by Uzma, 4 speakers, 1 topic. And I think something that I'd like to highlight as the good practice, you very quickly emphasized at the beginning of the presentation what your key talking points were. That was on the risk assessment profile. One point for improvement I would make as a recommendation is when you did your introduction, you gave a definition from the BBML. So if you could put that on a slide to show the document, the definition, that would be helpful. There was a bit of hanging time between a slide that didn't move and as you were speaking. So that would be helpful for future. I think one thing for me that came out as, again, Uzma mentioned, this competency is 7 hours. You had a slide presentation of 13 slides, assuming that your session was, say, 1 hour. I think it was probably a good mixture between a short, succinct presentation, which would then be followed by whatever activity that you would have chosen. So I think a good balance of time between speaking and then time for practice and a group activity. So I think for me that was also well paced, well done, and a good explanation. And another point that I picked up where you could have added a little bit more examples was during Emmy's section when you were on, I think, step 2 or 3. There was one example given, but I think you went through 3 different processes or 2 different processes, adding in an additional example as you pace your slides would have also been a bit helpful. But I think overall, very good job. Everyone spoke clearly. It was easy to follow. And I think very good adaptation of the slides and an excellent example of a disease that's of priority for Indonesia. So with that, I think congratulations to the team, and we will move on to our next speakers. So if the team is ready from Malaysia. And you're also getting congratulations from Tanya Pon in the chat. Thanks.
Good evening. I'm Salvanison here from Malaysia. Can hear my voice?
Yes, clear.
Yes.
Thank you.
Okay. Good evening, all the participant and commentator, and also the experts. Malaysia, we're going to present on equipment and consumable. Our team is from National Public Health Lab, which is Puan Hazli or Miss Hazli from food section, food division, and myself from clinical division. Another person is from Nahrim. Nahrim is a National Water Research Institute of Malaysia from Energy Transition and Water Transformation Ministry. So, okay, go back. This priest. Okay. After completing this module, participant will be able, first is they able to explain how equipment purchasing procedure are carried out in Malaysia especially. And then they also able to explain the criteria required for selection of new equipment, because Malaysia, they have a standard procedure to follow, especially on government sectors. So this will they will understand better the purchasing equipment, and then development preventive maintenance plan and equipment commissioning plan. This also we have standard policy and procedures, especially for government and other agencies, government agencies. And then last least the characteristic and inventory management system. Okay, this module will cover selection and acquisition of laboratory equipment, equipment calibration, prevention, maintenance of equipment, decommissioning equipment, and inventory management. Okay, if you see here, under the quality management system, out of 12, two important subject one is equipment management, another one is supplier and inventory management system. Okay, we come to purchasing of equipment. This is our main topic. Okay, before we start giving a lecture, actually we want to start with a group activity. Okay, why we start group activity before lecture is because all the leadership training program is the person that already experienced 5 to 10 years or 15 years in the laboratory, which they have done purchasing, they have done the calibration, everything. So that's why we want to give a small exercise to them. That's to discuss how the process, the purchasing process is practiced in their institution based on the value of the equipment itself, the price of the equipment. For example, we can give a biosafety cabinet, which is the price is 25,000, and a Malaysia ringgit freezer maybe a 52,000, a real-time PCR 200,000, and up to the million gas chromatography. Because in Malaysia, the prices determine the process of purchasing. So once they have done this exercise, and then we try to get what knowledge they have from the sharing of this group activity. Then only we go to our process. So like in Malaysia, we can divide into few categories. First is like direct purchase, as this is not only equipment for the equipment, but also applicable for all other reagent and consumables, which is if less than 50%, Actually, we go for direct purchase, which is usually involve three quotation or three vendors quotation and open to all suppliers. Anybody can come in as long they are registered with the government. And then for 50 to 500,000, they go for small process of tender, which is the advertisement is for 7 days. And usually we put in the notice board and we can call at least 5 vendors, which is familiar with that particular equipment or supply to come in. And then we have a term of Bumi Putra, which is a majority Malaysian Bumi Putra, especially like Malays and then Sabah Sarawak natives. Actually, this is to provide some facility for them. Those actually from 50 to 100,000. So, which is a small traders. And then usually we have 5 committee members or committee teams involved. First is a spec team, which is they will prepare the spec, which contain 3 or 4 person. And then we have another committee which will open up a document. And then we have a technical committee, practically from the laboratory personnels, and a price also usually from the laboratory. And finally, the decider is from the top management. And then we also have another bigger tender, which is more than 500K. They will advertise within 14 to 6 months, depend on the type of purchasing or the procurement, especially usually 6 months if involve any renovation or may involve some works. Okay, and it will be posted in a portal, online, and local broadcasting newspaper, all that, and it's open for all vendors. Also same, there are 5 committees, spec committee, and the document opening committee, technical committee, price and decider committee. And then there's another process of purchasing, which is a direct negotiation, which is usually it happened during emergency, like for example, during the COVID-19, it's happened. Direct negotiation, I have involved also in this the purchasing, and there's no limit. We just can call one company and usually have only one committee, which is appointed by a higher authority, and then we can negotiate price with them. Then we buy. We can, because sometimes we have a demand on the requirement, so we have that committee, which all these are legal process. Then we come to next selection and acquisition of laboratory equipment. Okay, selection and acquire equipment. Things to consider. First is the performance specification. This is very important because we know laboratory equipments are very high-end equipment. So the performance is very important, and then the application to the different field of the laboratory in one health approach. So the medical may need a different specification, and maybe in the science technology they may need a different specification. Then cost of acquisition, operation and maintenance, and reagent to be used. Either is a closed system, open system, for example, the serology or ELISA machine or examples. So we want to see also either it's a closed system, open system. Usually closed system reagent is very expensive, so that we try to go for the open system. But the open system that also sometimes have some issues. Okay, and then the equipment lifespan, that's very important when we prepare the specification, equipment lifespan. Then local service available. Some equipment may not have a local service available, so they directly purchase from overseas, and then there's no engineer, local engineer, no local technician to maintain the equipment. Then in our government, we have a government asset management, which is the procedure for acquisition of a new equipment, including market survey, procurement, registration, commissioning, use, storage, maintenance, and decommissioning. So actually we have, as I said just now, we have the document that's control the procurement of equipment for the government and government agencies. In fact, many private sectors also follow this procedure because it's very clear and easy to follow. So private sectors also follow these procedures. Okay, choosing a new equipment. Okay, establish a small, usually what happen, establish small work group for the specification committee. Then they must know intended use, define the analyte or target or purpose of the test, or diagnostic research or research whatever, and screening, and the population to be tested, and if it's involved samples, okay, what type of sample to be tested on that machine, because some equipment may not suitable for certain parameters or samples. And then the variable to be considered, the assay limitation, as I said just now, is it open system or closed system, safety and effectiveness, and finally is a budgetary requirement, include facility modification, need electrical, water, ventilation, temperature control, and cost estimate. Okay, and then we also must determine the cost per test, and then the long-term product availability and reliability, and then must do thorough literature review, because the technology is keep on changing. So when technology is changing, it's no point we're going to buy the old technology equipments, which is maybe obsolete in a few years' time. So that's we have to take consideration. And then the performance criteria, including sensitivity, specificity, especially if the equipment is directly test on sample or for the diagnostic purposes. And then reagent and shelf life, cost, availability, and chemical safety. Input from other knowledgeable stakeholders or colleagues. We can get review from other users. Usually that's why when we purchase equipment in the specification, we ask the vendor to specify who are the users using their equipment, and then write the specification and proposal plan. Okay, the specification criteria. Okay, usually there are this is not the whole thing. This is a part of it. Equipment match with the service provided. Okay, for example, the equipment, like for example, is a medical equipment. We must have a medical device authority approval. And for the others, maybe the same machine is not necessary. And then the performance criteria, the facility requirement, the cost, the reagent involvement, ease of operation, user training. That's another important point that we found is not stated in the presentation. I added those are in the red. Actually, the Malaysian team have added in the presentation. Then warranty, availability of manufacturer's technical support, service contract, so location in the laboratory, available space, accessibility, safety, and some machine we need to have a data management, but everything's now is online. So that the migration of the data from the machine to our laboratory LIMS lab information system, so which is very important. Some machine may need that. Okay, query equipment and documentation. Okay, equipment can be purchased or lease or rent, either through central acquisition or single or bulk procurement, especially during COVID, we buy in a bulk procurement. And then a region rental agreement. Malaysia also have a region rental agreement, which is especially for those equipment, the technology changes is quite fast. And then also can be donated by the certain non-government organization, which is they don't have any interest of conflict, and this must be get approval from the Ministry of Health Secretary General. Then condition of contract, including initial orientation and training for laboratory staff as part of installation. If computer or LIMS interface is required, include the purchase cost and if allowed. And then contract consideration, we also require parts manual, installation, proper installation, operation manual, service manual, warranty period, and content of service contract. Okay, installation and registration. Prior to installation, verifies physical requirement have been met, especially the brand, the model name. There's in the contract they must they provide the same equipment, safety check, electrical system, space, ventilation, water supply, ambient temperature, etc. Assign responsible person to oversee installation. Like in Malaysia, every department they have appointed asset officer. At least one person be appointed as asset officer in every institution. And then we also have a system which is a government management system, or in Malay they call it System Pengurusan Aset, or SPA. This is not a spa that they gonna massage all that. This is a SPA, that's like government management system. And then and every equipment that we purchase need to register under the SPA system, and then they will produce a birth certificate. So usually the asset officer in charge will produce a birth certificate for the equipment with the with a unique code number. And then also they produce a maintenance sheet, individual asset files, this to be maintained until the decommissioning of this equipment. And then especially in Ministry of Health, we have a consignor company that been appointed by the Ministry of Health to maintain this equipment, especially preventive maintenance, calibration been scheduled by them and maintained by them. Then for next is installation and testing and commissioning. We assign responsible person to oversee installation. Upon receipt, verify package content, verify package is not damaged, do not attempt prior to proper installation. If required, ensure the equipment is installed by the manufacturer itself. And we have to do a test performance and then user training. Incoming inspection. After a device is installed, we have to conduct incoming inspections to establish the inventory record, follow the manufacturer instruction, carry out an electrical safety check, conduct an operational check, perform initial calibration, determine its frequency of outline calibration. And we also need to do a function check, monitor equipment parameters periodically, daily. This is a role or responsibility of the laboratory personnel to do daily, weekly, and monthly observation and record the parameters. And then after major instrument repair, they have to, before we use the equipment, we have to monitor. And after power fluctuation, if required, it's not used for a period of time. Then maintain instrument maintenance log, like for example, equipments that need to monitor the temperature, especially incubator, freezer. We need to monitor the temperature, and maybe like CO2 incubator, we have to monitor the CO2 gas too, and wavelength calibration and autoclave temperature chart for the autoclave. Okay, validation, two options to evaluate performance characteristic, comparison of result between new and method and reference of gold standard method, evaluate result using new method, verify materials, reference panel, and often available commercially. Okay, we come to the next part, which is calibration. Okay, when to calibrate equipment, usually as recommended by the manufacturer, we have to do calibration once a year or 6 months once. And then the calibration should be done using a standard method. Okay, how to calibrate a reagent. So some equipment, maybe we need a reagent to calibrate. So the reagent, maybe we can get from the reference material, or we can make our own reagent as long we follow the standard. Then perform initial calibration after reception of a new piece of equipment, use calibrator or standard, follow manufacturer instruction, determine frequency or routine of calibration. As I said just now, maybe so once a year we need to calibration, or after the equipment is breakdown, or for long term the equipment not be used, we have to do calibration. And then acceptance criteria should be established in advance in the laboratory quality documentation. Okay, how calibration may affect the result. If run fail at QC and at EQA, the result cannot be delivered. The consequences result cannot be delivered. And if the analyte is undetected, it can affect the position of the result, the erroneous of result. So this is the laboratory management should understand well about this process. Okay, and for this calibration, we like to give one small group activity. Okay, this is work with the group to discuss what calibration policies currently being used in their laboratory, and then share what works and what doesn't, and then be prepared to share the group inside. Okay, next is the preventive maintenance. Okay, preventive maintenance is a routine cleansing, adjustment, and replacement of equipment's part. Okay, we have a chart actually for the maintenance plan. For each piece of equipment, established routine maintenance plan to include: one is the frequency of the all maintenance tasks, function check, routine replacement of parts, and like for example, in the Ministry of Health, the concessionaire company will record every single ppm, and then they will also calculate the cost of maintenance. This is to determine at the end of the service or the decommissioning, is it worth to use that equipment, or we can decommission because the cost of maintenance is sometimes higher than the purchase price. Okay, implementation and maintenance plan, we assign a responsibility. Then develop document policy and procedure is very important. Then maintain records, and finally train the staffs. Keep a dedicated logbook for equipment or required equipment records. Okay, every single equipment that we purchase under the guideline or a document that produced by the government, they have a few we call it QPA forms, which is we have to record every single work we have done or we have on that equipment. For example, the routine maintenance, the calibration, service repair by manufacturer, all problem, and also the movement of equipment, the record. Sometimes, like small equipment, the vendor may take to their factory or to their office to repair. So we have to record every single movement of the set, and then the function check, and usually we keep the log close to the equipment. Then we also need to create an inventory log, water record, usually instrument type, model number, serial number, and a location in the laboratory, date purchase, manufacturer and vendor contact information, warranty, expiry date, and spare parts too. Everything will be recorded in that asset system. Then finally, decommissioning of equipment. Okay, what is decommissioning equipment? The action taken when equipment will not be used for testing and is removed from active service. So usually occur when the equipment is usually not functioning, and then low testing capacities. Now with the change of new technology, we may find equipment that can the capacity is a double or triple of the previous equipment. For example, a good example I can say is during COVID. Previously, we had one extraction machine which can prepare 24 extraction in 4 hours. So after the COVID, we find that within half an hour we can produce 96 extraction. So, and that's